Welcome to the Unit 3 material for MLT 110. This will be part 1 of the lecture. In this unit, we'll be discussing immunohematology and immunology. Immunohematology is the branch of immunology that deals with the immunologic properties of blood. Also in this unit, we'll be covering the very introductory material of blood banking. In part 2 of the lecture, we'll be discussing ABO testing, which is basically blood typing, and also antigen antibody testing. The study of immunology can bring together a wide variety of disciplines, and some of those disciplines can include microbiology, oncology, allergy science, lab science, genetics, transplantation medicine, among many others. Allergic reactions are just exaggerated immune responses to allergens, and some examples of those would be a reaction like hay fever or a poison ivy rash. An autoimmune disease or reaction is when the immune system attacks itself or its own cells or tissue, and an example of that would be rheumatoid arthritis. If a person was to have a deficiency or a malfunction of the immune system, this could allow cancer cells to grow because cancer cells are cells that are growing out of control. And the person's immune system is not in place to stop the growth of the abnormal cancer cells. The immune response is made up of a combination of tissues, cells, cell products, and biochemicals that interact to produce the overall immune response. The body's immune system can provide a defense mechanism against foreign substances, and foreign substances are referred to as antigens. And antigens can come in several different forms. They can be molecules or viruses or blood cells, tumor cells, bacteria, and fungi. The defense mechanism that the body will produce to fight against antigens are antibodies. For a substance to stimulate an immune response, it must be antigenic, meaning it must contain a protein or a carbohydrate. The small portion of the molecule that is actually antigenic is called the epitope. So let's look at the image over here on the slide to look at what an antigen looks like and what the epitope looks like and what the antibody looks like. The antibody is usually illustrated in a Y shape. And remember, the antibody is what the body produces to fight against the foreign object. The antigen is usually the foreign object, and the epitope is where the two are going to attach, the antigen and the antibody. There's some different types of immunity that your body has. The first type is natural resistance. This is also referred to as innate resistance. The response is nonspecific, so there's no previous exposure required for your body to respond. The body has a variety of ways in which it can react or protect itself. And some examples for natural resistance include just your skin, mucous membranes, stomach acid, even enzymes in tears and sweat, and also your phagocytic cells like your white blood cells, your neutrophils, and your macrophages. So I always like to refer to your natural resistance or your innate resistance as your first line of defense. Next up is specific immunity. This type of immunity can actually recognize and remember different antigens, and it can respond to a specific invader of the body. So let's talk about some different characteristics of specific immunity. First up is recognition. The body or the immune system can recognize small differences in antigens in the environment, and also the immune system has the ability to distinguish the difference between antigens and yourself or your own body. There's also the characteristic of specificity, which is the ability to respond towards a specific antigen without reacting to other similar antigens. The last characteristic is memory. This is the ability for your immune system to remember an antigen long after the initial exposure. This is also referred to as the amnestic response. And this is the basic idea behind immunizations. So let's say you go get a vaccine. So the patient is being exposed to a foreign antigen through receiving the vaccine. So the patient's immune system is able to mount a immune response because they're being exposed to that vaccine. And because that person has had a immune response as a result of that vaccine, the body will then be able to fight off that invader if they are exposed to a real disease. Lymphocytes, which is a type of white blood cell, plays a very important role in bringing about a specific immunity response. On the previous slide, we said that lymphocytes play an important role in specific immunity. And with knowing that, there are two different types of specific immunity. There's humoral and then there's cell-mediated. And the two different types are differentiated by the type of lymphocyte that respond to the antigen. So let's look at our two types of lymphocytes. We have our T lymphocytes that are referred to as T cells, and we have our B lymphocytes that are referred to as B cells. The B cells tend to attack invaders on the outside of the cell, and T cells tend to attack invaders on the inside of the cell. Let's look at humoral immunity a little bit more in detail. And remember, our humoral immunity is in relation to our B cells. B cell humoral immunity provide protection against things like bacteria, toxins, and antigens. Also, B cell humoral immunity produce antibodies to fight against antigens. And also, B lymphocytes have been converted to antibody producing cells called plasma cells. Let's look at the image on the right hand side of this slide. This is a flow chart of the maturation sequence of your blood cells. Let's start up here. This is your blood stem cell where all your blood cells start. And it divides up into your myeloid stem cell and your lymphoid stem cell. We're more interested in particular on your lymphoid stem cell side for this particular discussion. And it goes down to your lymphoblast and then to your B lymphocyte and your T lymphocyte. These are the two that we're interested in for your specific immunity. And on the slide that we were just discussing humoral immunity, your B lymphocyte can ultimately turn into your plasma cells that are antibody producing cells. And just for reference, this is what a plasma cell can look like under the microscope. This right here is your plasma cell. This is a stained slide, so that's why it has some color to it. This was what it would look like under a microscope. And just so you'll know, we'll be learning about this whole maturation sequence of blood cells in hematology, MLT 120. Next up is your cell-mediated response or immunity, and it's associated with your T cells. Remember, this is still under the specific immunity umbrella. Just like the humoral immunity, remember humoral's associated with B cells, cell-mediated associated with your T cells. In the cell-mediated or T cell immunity, the T cells protect against things like viruses, fungi, tumor cells, or intracellular organisms, so inside the cell. Foreign invaders are presented to your T cells by your macrophages or your dendritic cells. Your macrophages has the ability to phagocytize. So what does that mean? It has the ability to engulf foreign objects. So it can take viruses, fungi, whatever, into its cell. It can engulf those foreign objects, and then it can take those foreign objects within its cell the cell itself, and it can present those foreign objects to the T cell. T cells can secrete something called cytokines. Cytokines are a small molecule that can communicate with other cells, and you can kind of think of them as little messengers because they can work with other cells to help take out foreign invaders and to help regulate the immune system or the immune response. Let's talk a little bit more about antibodies, and another term or name for antibodies is immunoglobulins. So what exactly is an antibody? It's a protein produced by a plasma cell. Remember, the plasma cell is the result of a B cell or a B lymphocyte. Remember, we looked at the maturation sequence of blood cells on a previous slide, and it comes from the myeloid line. So antibodies are secreted into body fluids in response to the body being exposed to an antigen. Foreign antigens in the body bind or react specifically with antigens at the site of the epitope on the antigen. You can think of it kind of like a lock and key system. Antibodies are produced by the body for a particular antigen. So just not any old antibody is going to fit any old antigen. And again, they're going to be connecting at the epitope on the antigen. So if you look at the image, this particular antibody was made by the body for this particular antigen. You see how it was made to fit together, so lock and key. So this particular antigen is not going to work for this particular antibody. You see, this is not going to fit in here. And again, this is just an illustration. So antibody, antigen are specific for each other, lock and key. We have 5 different classes of immunoglobulins or antibodies. IgG is up first. It's the most abundant type of antibody in the blood. It's a monomer, meaning that it's a small sized antibody. It remains in the blood for a long amount of time, providing long-lasting immunity. Newborns are born with this type of immunity because it passes from the mother across the placenta to the baby, and it gives the baby immunity for several months. This is known as passive immunity. Next is our IgM antibodies. It's the largest. It's a pentamer, and it's the first class of antibodies that's produced after birth by our newborns, but it does not provide long-lasting immunity. Next is IgA. It's the second most abundant in blood. It's a dimer. It's usually found in secretions from the respiratory and intestinal tracts. You usually find it in things like tears, saliva, breast milk. IgA in breast milk gives passive immunity to our newborns. IgD, we only have small amounts of IgD in the body, and little is known about its function, but it is a monomer. Okay, finally is IgE. It is a monomer, and it's involved in allergic reactions and parasitic infections. It's the least common one in our blood. It's known to cut eosinophils, basophils, and mast cells, and these are all white blood cells. Let's look at this chart to understand the different types of acquired immunity. First off, what is acquired immunity? It's immunity that develops over your lifetime. You have active immunity and you have passive immunity. So let's first look at active immunity. Active immunity develops in response to an infection or a vaccine. And then passive immunity develops after you have received antibodies from someone or you've received antibodies from somewhere else. So let's go back over here to active immunity and look at the 2 categories under active immunity. You have natural and you have artificial. So let's look at natural. So active immunity, natural. You have antibodies developed in response to an infection. Artificial, you have antibodies that develop in response to a vaccine. So in natural, you've been exposed to something in the environment and you're naturally developing antibodies. In artificial, you receive a vaccine and your body's developing antibodies as a result of the vaccine. Okay, over here on the passive side, it's also broken down into natural and artificial. So what would be an example of getting natural immunity on the passive side? An example would be getting or receiving antibodies from a mother to a baby through breast milk. Baby is passively receiving antibodies from the mother. On the artificial side, you would be receiving antibodies through some kind of medication. And examples of getting those antibodies through medication might be through gamma globulins or some kind of infusion. Okay, within our immune system, we have two types of antibody responses. We have a primary response and we have a secondary response. The primary response is the body's response after the first exposure to a foreign antigen. So usually the body produces IgM antibodies, and the response time for the IgMs is usually slow because the body system has to learn the antigen because they've never been exposed to it before. So we think of the turtle. The IgM is the turtle. It's slow. It's learning the antigen. The secondary response, or the amnestic response, is the response after the initial first exposure. And the secondary response is going to produce IgG antibodies. This response is much faster because the body remembers the antigens from the first exposure. And we're going to think of this as the rabbit because the rabbit is much faster. I mentioned this a little bit earlier on in the lecture, but autoimmune responses is when the body reacts or attacks its own self or its cells or tissues. Some examples of conditions where this happens is rheumatoid arthritis or lupus, diabetes mellitus, multiple sclerosis, or thyroid disease. The links where you can check out more information on each of these diseases are under Unit 4 on your Moodle page. Now let's talk about immunologic testing. Antigen-antibody reactions. An antigen can be detected in a patient sample by using commercially prepared antigen-specific antibodies. Antibody reagents can either be monoclonal or polyclonal. So what's the difference between the two? Monoclonal antibodies use one class or one specificity. So you're going to only have a reaction with one epitope. And these are produced in labs and are used in many test kits. So let's look over here at the image of a monoclonal antibody antigen reaction. So one antigen is only reacting with one antibody. So you only having one epitope reaction. So with polyclonal, you're going to have a mixture of antibodies produced by greater than one antigen cell line. So you have your antigen here in the middle, and you have multiple different antibodies that can attach to it. We know these are different antibodies by the different color epitopes that are attached. Where this one over here, you just have this little blue attachment. But here on the polyclonal, you have different colored attachments, so we know it's a mixture of multiple antibodies that's attached. So this is polyclonal, multiple antibodies, monoclonal, only one. The next couple of things you want to think about when you're selecting a test is the test specificity and sensitivity. First off, sensitivity is the lowest limit that can be detected. So a lower concentration capable of being detected by the test method. If there's a failure for a test to detect a low amount of a substance, it can result in a false negative. Let's say there's a very low concentration of a substance in a sample, but the test is not sensitive enough to detect that low amount and it comes off negative. That would be a false negative. Specificity is the ability of a test just to detect the substance for which the test was designed for. If a test has a reaction with other substances, it decreases the specificity. So that could be a false positive. This concludes part 1 of our lecture. I'll see you over in part 2 where we'll get more into blood banking.